Today's session, we're looking at the overall factors that affect the rate of photosynthesis. So basically, how well photosynthesis can be done, at what rate it is being done. So yeah, I guess this is a good chance for me to quickly go through. So like I said, right, CO2 is the proper substrate that Rubisco wants. Oxygen is the inhibitor molecule. When oxygen binds, it promotes photorespiration. If CO2 binds, that's going to promote photosynthesis. So two reasons, sorry, one primary reason that can cause Rubisco to allow oxygen to bind to it is a hot and dry environment. Okay, when it's hot and dry, the plant loses more water, and because it's losing more water, it needs to preserve its water content, so it closes its stomata. When it closes stomata, there is no gaseous exchange occurring, so carbon dioxide isn't being taken up, oxygen isn't going to be released from inside the cell, inside the plant. So that means there will be suddenly a high concentration of oxygen within the leaf where photosynthesis is occurring. And like I said, with substrate concentration, if there's more inhibitor molecules present, then they will more likely bind to the Rubisco. And like I said, that will cause photorespiration, and we do not want that. Primary reason is photorespiration takes energy, photosynthesis gives energy to the plant. Okay, so essentially, if it keeps doing photorespiration, it's basically you going for a 1 mile run while you're still hungry. So you're using your energy when you need energy, and eventually that will actually cause the plants to die if they can't stop doing photorespiration instead of photosynthesis. Another thing that can cause this to happen is when it's a hot environment where Rubisco's the enzyme's affinity to oxygen increases, meaning that it's basically more attracted to oxygen when the temperature is higher. So essentially, that's why in a hot and dry environment, plants tend to die fast, especially C3 plants. These are C3 plants that don't have the ability to counter-react photorespiration. So whenever they're put into a hot and dry environment, they will experience photorespiration because of the stomata's closing, oxygen building up, and also because Rubisco's affinity to oxygen increases when there is a hot environment. So when the temperature increases, Rubisco likes oxygen more, so oxygen will bind to Rubisco more, and photorespiration will be increased. Okay, always remember this: photorespiration is bad. We don't want it. In fact, plants want to get adaptations to make sure they minimize photorespiration. That's where C4 and CAM plants come in. C4 and CAM plants, basically, the biggest thing to note about them is that they have adaptations to minimize this photorespiration. Okay, I'm going through this very quickly because this is an entire topic by itself. I don't think I can cover it properly like this, especially what C4 and CAM plants are. You guys will have to go through that on your own. Okay, so today we're gonna look at what are the factors that affect photosynthesis. So essentially, what could make photosynthesis work better? What could make it work worse? There's light availability, there's water availability, there's the temperature, there's carbon dioxide concentration, and these factors each affect photosynthesis differently in how it affects it. So this time there's no glossary task for you guys because pretty much all the words that you're looking at in this topic have already been covered in enzymes, basically enzymes. So like I said, enzymes play a huge role in photosynthesis. So when we're talking about factors that can affect photosynthesis, we are inadvertently talking about factors that affect enzymes. Okay, as you can see in the next slide. So obviously, with photosynthesis, light intensity is a huge factor to play when it comes to the rate of photosynthesis because light availability directly influences the first stage of photosynthesis, which is the light dependent stage. So obviously, if it depends on light, it needs light, so that affects the photosynthesis. Temperature, like I said just now, temperature directly influences the affinity of Rubisco. If Rubisco is present in a high temperature, then it will have a higher affinity towards oxygen, which is the unwanted inhibitor molecule. pH and temperature both can be put into the same category because they're both affected by enzymes. Sorry, they both affect enzymes. So essentially, these two are also talking about the enzymes and how they can get affected within the plants. Carbon dioxide, basically substrate concentration, another one of enzyme factors. So carbon dioxide here, but essentially that's substrate concentration because that carbon dioxide is what the enzyme wants. So here it's a substrate concentration. Water availability, obviously, and enzyme inhibitors. Water and carbon dioxide both are prerequisites. They're reactants required for photosynthesis to occur. So that's also another reason why they're here. And with enzyme inhibitors, obviously, if any enzymes are present, their activity can be their activity can be influenced by the enzyme inhibitors. So they play a role in the rate of photosynthesis as well. One thing to note is that between the different types of plants that do photosynthesis, C3, C4, and CAM plants, each of these factors play differently in the sense that in a C3 plants, temperature or light intensity might have a higher influence compared to in C4 or CAM plants, or it can vary between each of them. We'll go through how each of them is different when we are covering it. One thing to note here in for your exams, right, is that they will often ask questions from this topic. It's a very hot topic to ask. Factors affecting enzymes, sorry, factors affecting photosynthesis. One way they would actually ask it is that they would use a graph and then ask questions based on that graph. So when you're looking at a graph, it's very easy for them to try to confuse you guys by using different axis titles, right? For example, they sometimes can use light intensity, and then on the y-axis they use photosynthetic rate. That's a pretty obvious one, right? Photosynthesis rate and how it's affected by light intensity. But they can also change it up, say relative rate of photosynthesis over temperature. So you would need to be able to associate the idea that if they are saying anything related to photosynthesis and anything related to the factors affecting photosynthesis, that it's about that topic. Okay, so it could be temperature, amount of carbon dioxide present, light intensity, relative light intensity, any of those factors that we just looked at could be down here in the y-axis. As long as the y-axis says anything related to rate of photosynthesis or absence of photosynthesis, you need to be able to relate it back to this topic. So you can see here, you can come across different axis labels that represent factors affecting the rate of photosynthesis. Light can be represented as light intensity, availability of light, absorbed light, simply just light as well. It could be just the word light, and you need to be able to understand that. It's also the same with your y-axis, but with your y-axis, it is a bit more obvious just because they have to mention photosynthetic or photosynthesis when they're putting the y-axis. So you could say that. Another thing that they could put in the y-axis is they could say something like how much glucose is produced, right? Because glucose is tied with photosynthesis. It's a product of photosynthesis. So if they say concentration of glucose in the plant, and you can see it increasing, that is a sign that photosynthesis has been occurring, right? So you need to be able to relate the ideas you've learned, relate it back into this graph and understand what is it that they're trying to test here. Anyway, moving on. All right, the first factor affecting photosynthesis, light intensity. Essentially, light intensity is most important in the first stage of photosynthesis, which is the light dependent stage. Why is it important? Is that the energy gained from the light will be essential for the production of ATP, and the splitting of water molecules is also essential for the electrons to move across the electron transport chain to power the hydrogen pump as well. So there is a lot of functions that the light serves, but essentially without light being present, it will not be able to, the first stage of photosynthesis will not be able to be carried out. In fact, at night, light intensity becomes a limiting factor. Does everyone remember the phrase limiting factor? Is anyone confused by the phrase limiting factor? Now's your chance to clarify. Right? Limiting factor basically is the factor that makes sure the reaction rate can't go up anymore. So if you see here, you can see light intensity is increasing, rate of photosynthesis is increasing until it starts to plateau. Right? Basically, no matter how much light you give it after this, it won't increase the reaction rate because some other factor is acting as the limiting factor to make sure the reaction rate doesn't continue going up. That could be carbon dioxide availability, that could be water availability, it could be any other factor, it doesn't matter. But the fact is, you see the reaction rate plateauing because some other factor is holding back the reaction rate. That is basically limiting factor. What is holding back the reaction rate from continually increasing? That's how you would explain a limiting factor. So if you ever see a graph like this and they ask you to explain why does the graph plateau, you would essentially say that light intensity has reached a saturation point or saturation curve, and some other factor has become the limiting factor. So you could say something like carbon dioxide availability or water availability. Like I said, any of the factors can be used, just not light intensity, because they've shown you here that light intensity is increasing. They keep increasing the light, but the reaction rate is still consistent. So something else is holding back the reaction from actually increasing the reaction rate or the rate of photosynthesis. Okay, think of it like this. If you have a recipe to make cake, even if I give you 10,000 kgs of sugar, you can only make enough cake with the rest of the ingredients, right? If none of the other ingredients are readily available, you can just make enough for one cake. You can't make a lot of cakes. Even if you have all the sugar in the world, you need the other ingredients to be present as well. Then only you can make a lot of cakes. Okay? If you have a lot of sugar, but the rest are limited, you can only make as many as you have. Right? Same thing here. Even if you give a lot of light for the plants to do photosynthesis, if any of the other factors that we looked at are not at the same level, then they will become a limiting factor because they limit the rate of reaction. All right. So like I said here, right, the reaction rate is going up and then it starts to plateau. This is where it's considered a light saturation curve. Basically, the amount of light you can give the plant and for it to carry out photosynthesis has been saturated. No more benefit from giving any more. This is because the maximum possible rate increases as the enzymes within the chloroplast are operating at their full capacity. Basically, all the chloroplasts have absorbed the necessary sunlight and they can't absorb anymore because all the enzymes are already working at full capacity. So light, however, influences the flight text in the same manner. Essentially, C3, C4, and CAM plants, they all require light in the same level. So light intensity would be considered one of those factors that are equally affecting all three plants because that's how imperative light is for the entire process of photosynthesis. Because the first stage of photosynthesis is the same between all three plants, C4, C3, and CAM. So essentially, if light can't even activate that first stage, all three plants will be affected. So here's a graph. At this stage right here, there's no light, so the rate of photosynthesis is at 0. So light is the limiting factor here. The fact that light isn't present means that the reaction rate can't go up. But once you come here, this is considered the light saturation point because no matter how much light you add after this, it won't increase the reaction rate. So why? Another factor has become the limiting factor. So any factor you can give would be accepted because any factor could be a limiting factor here. They're not giving any other information. You could even say that the enzymes are the limiting factor as well because enzyme availability has reduced to 0 because all the enzymes within the chloroplast are operating at full capacity. So enzyme availability becomes a limiting factor as well. So essentially, when you think about light intensity, we are talking about visible light, right? The light that we get from the sun is considered visible light. But when we basically think about it in terms of physics, light can exist. So light can exist in a spectrum of colors, and the different colors affect photosynthesis differently. And you can see that in this graph here, especially in terms of green. Green always affects photosynthesis at a very low, relatively low level. So essentially, the rate of photosynthesis will drop when the color that the plant is exposed to is green. This is unlike violet and red, where the greatest rate of photosynthesis can be seen. This can actually be tested using an experiment. But essentially, the two colors that increase rate of photosynthesis are violet and orange, and the color that reduces the rate of photosynthesis is green. Okay. So the next factor we're looking at is temperature, and temperature can temperature can have an effect on photosynthesis in the same way that it has an effect on enzymes, right? Essentially, other than the Rubisco, temperature also directly influences all the other enzymes in the plant. So optimal temperature and then any other temperature will reduce the enzyme activity. And if you go above the optimal temperature too much, obviously enzymes denature, lose conformational change at the active site, and it loses its function. If you go below the optimal temperature and keep going, again, enzymes freeze, they won't be able to activate. But like I said before, any frozen enzyme can regain its function if you just heat it back up. But when it comes to going above the optimal temperature, obviously the enzymes will not be able to regain their function even if you go back, because once the protein loses its original shape, it cannot gain that shape back. It is already permanently deactivated, right? So all plants have their own optimal temperature in the same way that I said all living things have their own optimal temperature, right? Based on the living thing's optimal temperature, it will have enzymes that are designed to withstand those temperatures. So it's not going to be fixed. No plant enzymes are going to be, some plants can have a very different optimal temperature versus other plants. So you can't say that it's fixed, but you can always refer to the optimal temperature and saying that if they go lower or higher than the optimal temperature, what can happen? Okay, unless if they already gave you the actual optimal temperature range, then only you should use that reference. Okay, say when the temperature goes above 35 to 38, then what happens? But this is only if they've provided you the optimal temperature. If not, just use the word optimal temperature, because you can't assume what the optimal temperature of a living thing is unless we know the facts. So moving on. Okay. So essentially, the optimal temperature and pH values and ranges may differ between C4 and CAM plants and C3 plants. So you would have to pay attention to the plant before you can assess what the optimal temperature is. Like I said, permanent loss of temperature if you go above, enzymes are frozen if you go below, enzymes can regain function if you unfreeze them, enzymes cannot regain function if they become denatured. Yeah. Moving on. So next factor we're looking at is pH. And pH affects the enzymes involved in photosynthesis, where if the pH of the environment goes above or beyond the optimal pH, the enzyme will automatically denature. So same with the enzymes that we learned before. With pH, it will cause denature whether it's going below or above, that doesn't matter. As long as it's not the optimal temperature, it will become denatured. And like we talked about, when it comes to denatured, what is happening? Denaturing means that there's conformational change occurring at the active sites of the enzymes, making them non-functional. Right? That's how you always explain that. Any questions about the stuff we've looked at so far? Any of the factors not clear? Same thing. So essentially, if this was 7, right? Not necessarily, it could be any other pH depending on the plant. But if this was 7, you by going lower, you're becoming more acidic. It can denature the enzymes. If you go above to a basic, it can denature the enzymes. So essentially, it's the same thing with temperature. Each plant will have its own optimal pH, but if you go below or above the optimal pH, then they will become denatured. Okay? Only with temperature, if you go lower than the optimal temperature, the enzymes get frozen and they can actually get thawed and regain their function. That's the only exception. Other than that, they usually get denatured if you go too high or if the pH is too low or too high. Yeah, like I said here, right? So this shows the optimal temperature range of a C4 plant versus a C3 plant. And remember the difference between, okay, you guys weren't here for this. The difference between C3 and C4 is the kind of environment that you can find there. C4 is basically plants that can survive dry and hot environments better than C3 plants. So these are your corn, your maize plants, some of your crops, and a few wheat grasses as well. These are basically have adaptations allowing them to survive in hot and dry environments, and that directly is a part of minimizing photorespiration. They are able to survive in a hot and dry environment because they have adaptations to minimize photorespiration, so they don't have to worry about being in a hot and dry environment. And that's why you can see that their optimal temperature is actually leaning towards hotter temperature than C3 plants because they actually have the adaptations that allow them to survive in that kind of environment. So obviously they prefer to be in that kind of environment. Right? So, like I said, when it comes to the rate of photosynthesis, you can see there's a sharp decline once it goes above its optimal temperature, and that's present with any enzyme. If it goes above its optimal temperature, denature, conformational change at the active site, and loss of function. So moving on, let's look at carbon dioxide levels. So how does carbon dioxide affect photosynthesis? Obviously, carbon dioxide is one of the reactants required for photosynthesis to occur. Just in that fact alone, it is obvious that it's a very important factor affecting photosynthesis. So when there is a higher concentration of carbon dioxide, obviously the rate of reaction will rise, and same with light availability, eventually it will reach a saturation point, right? And at which point the reaction rate will plateau. So this is essentially when every enzyme of Rubisco has bound to CO2. So essentially the enzyme availability becomes the limiting factor here. So with light, rate of photosynthesis will increase as carbon dioxide increases and will stay stable. Okay. If stomatas are closed or there is a low CO2 level in the atmosphere, the CO2 may limit the rate of photosynthesis. So essentially, if there isn't a lot of carbon dioxide inside the plant, then rate of photosynthesis will remain relatively low. But if there's a high exposure, then it will keep increasing until it reaches its saturation point, at which point something else becomes a limiting factor. Okay, this idea of another factor becoming the limiting factor is always going to be around for enzymes and stuff relating to rates of photosynthesis as well. You can always be able to use that answer of it's reached a saturation point for carbon dioxide where another factor acts as a limiting factor, and you can give an example. Here you could say that light intensity could be a limiting factor, or water availability is a limiting factor. Again, the factor that you use is not really too important because they haven't specified. But if they specify something being the limiting factor, then you have to use that. Okay. Okay. All right, Rubisco, like I said, why is CO2 necessary for photosynthesis is that it is used by Rubisco to initiate photosynthesis, specifically in the second stage of photosynthesis, the Kelvin cycle. And if there is a low concentration of CO2, why would the rate of photosynthesis decrease? Is because if there's a low rate of photosynthesis, then there is going to be competitive inhibition done by oxygen, and that means Rubisco will undertake photorespiration instead of photosynthesis, which is a wasted reaction. So essentially, if there's a low concentration of carbon dioxide, then the Rubisco will more likely bind to oxygen, which is going to trigger the waste reaction. So essentially, carbon dioxide is required for photosynthesis. If there's a lot of it, then the rate of photosynthesis will increase until it reaches its saturation point, and then some other factor will become a limiting factor. Yeah.
This photorespiration for wasted reaction.
B, yeah. So yeah, basically there's a lot of stuff in B that's going to be mentioned. So I would highly recommend you guys take your time, go through it yourselves. And if you still feel that you can't understand it, let me know. We'll do another lesson on that. It'll probably have to be the study session lesson because your SAC's coming up and you still have a lot of content to cover. All right. Between C4 and CAM plants, carbon dioxide level concentration is much more control, right? Carbon dioxide levels within C4 and CAM plants are controlled much better compared to C3 plants because it directly relates back to the adaptations that it has to minimize photorespiration. Okay, CAM and C4 plants are different from C3 because they have adaptations to minimize photorespiration. That is directly related to the carbon dioxide levels within the plant. Okay, how do they minimize photorespiration? Is making sure that they constantly have a good supply of carbon dioxide, unlike C3 plants who can't guarantee that constant supply. Moving on, right? So as I said here, this initial increase of rate of photosynthesis is due to the chloroplast having a higher concentration of CO2. So as the concentration of CO2 is increasing here, the rate of photosynthesis will increase along with it until the saturation point, and then another factor becomes the limiting factor. So this relationship is just like the relationship between substrate concentration and enzyme activity, because remember, carbon dioxide is the substrate for Rubisco to work. Moving on. So another fact is water. Obviously, water is an important reactant in the light-dependent stage where water is broken down to give electrons to the photosystem, and also it is where the oxygen comes from as a product of photosynthesis. So essentially, the breaking down of water both supplies the necessary things to carry out the light-dependent stage, and also supplies one of the final products of photosynthesis, which is the oxygen that we breathe. Oh, so this factor also influences the opening and closing pores of the stomata. So how much water the plant is able to retain directly influences whether it opens or closes its stomata. And this is only for C3 and C4 plants, because CAM plants keep their stomata closed all throughout the day and only open them at night. They are the only ones that directly control their stomata. Whereas with C3 and C4 plants, they open and close their stomata based on the amount of water that they have. If they have a lot of water stored within them, then they are more likely to keep them open because they can lose some water and still be fine. But if it's a dry environment and the plant doesn't have a lot of water and it's already losing so much water through its stomata, then obviously it will close it just to prevent more water loss. Essentially, if it's a good example of this. If you are in a desert and you are losing a lot of water, you would do everything you can to try to minimize your sweat, right? You wouldn't want to sweat too much because you're losing water already. So, well, the plants can by closing their stomata. We can't help it, but the plants can. All right. So yeah, this graph basically shows that, pretty much shows this, right? So let's go through the graph. Let's follow along, see what happens, right? So what happens when there's less water in the external environment? The plants will close the stomata to prevent water loss. If they close the stomata, this creates a problem where oxygen that's being produced in the light dependent stage can't leave the leaves, and the CO2 outside the environment can't go in to be used for the second stage of the photosynthesis, the light independent stage. And because oxygen is being built up, its concentration is increasing. And like I said before, Rubisco is going to bind, substrate concentration influences what Rubisco binds to. If there's going to be a lot of oxygen present, then it will bind to the Rubisco instead of CO2, and that promotes photorespiration. And then essentially, because photorespiration is happening, photosynthesis can't happen. So the plant is using up its energy when it needs more energy. And if this keeps happening, the plant will eventually die, right? So what can cause another factor that can cause the stomata to close is if the temperature suddenly went up. So essentially, not just about water around the surrounding. If the temperature suddenly went up, then the plant will start to lose more water through its surroundings, right? Essentially, if I suddenly raise the temperature in here to 30, 40 degrees Celsius, all of us would start sweating. Same thing for the plant. If suddenly the temperature went up, it will start to release more water through the stomata, which it hates doing. Okay, so if it does too much of it, it hates it, so it, closes the stomata, and again, it creates that pathway where photorespiration is occurring more and photosynthesis rate is actually dropping. Another thing is that if the temperature increases, it also denatures the enzymes. So that's another reason why temperature can influence photosynthesis rate, because two reasons why temperature has an influence is because temperature directly influences whether the stomata is opening or closing, and it also influences whether the enzymes are going to be denatured or not. Okay, so in summary, decreased amounts of water leads to closed stomata, and in turn, decreased CO2 and increased oxygen concentrations. This can be different for C3 and C4 plants because, again, C4 and CAM plants are not affected by water availability unless it is extreme, whereas C3 plants are more susceptible to water loss because C4 and CAM plants have built-in adaptations to reduce photorespiration. So they have systems in place to reduce water loss as well, especially CAM plants, which keep their stomata closed throughout the day when it is hottest. Okay, that is a major adaptation that CAM plants have that you need to mention if they ever ask. Okay, if they ever ask, what are some adaptations that C4 plants have over C3 or C4? There are two things you need to mention. One is talking about the thing that we covered in the previous lesson. I highly advise you guys to go check it out. Essentially, it's about when the carbon fixation step is done. When the carbon fixation and the rest of the Calvin cycle is done. The carbon fixation in CAM plants is done during nighttime, and then the rest of the Calvin cycle is done during daytime. That's the first factor. By doing that in that way, it can minimize photorespiration. Another factor of why CAM plants can efficiently do photosynthesis is that they keep their stomatas closed during the day and only open them during the night, which essentially reduces the water loss that they experience. And last factor is enzyme inhibition. So we've talked about a specific enzyme, which is rubisco, and how it can be inhibited through oxygen, and how it actually wants to bind to carbon dioxide. So in this example, competitive inhibitors are those that bind to the active site of the enzyme. Essentially, that's talking about your rubisco, right? Rubisco is an example of competitive inhibition because the oxygen molecule binds to the spot where carbon dioxide wants to bind to. So it's essentially blocking the binding spot. Another type of inhibition is non-competitive inhibition, which basically changes the active site by binding to another place in the enzyme called the allosteric site. And when it binds to the allosteric site, it will essentially reshape the active site, causing it to become non-complementary to the substrate. So remember, with enzymes, it's all about the shape and the structure. Okay? If the shape do not match each other, if they're not complementary, then it won't be binding together. So that's what's happening here. The inhibitor molecule is essentially changing the shape of the active site such that it is no longer complementary to the inhibitor. So there are very different inhibition methods. One is binding to the active site and blocking it. The other one is binding to the enzyme and then changing the active site to become non-complementary. Right? And this can also be done reversibly or non-reversibly. Essentially, to know whether it's reversible or not, you need to expose the enzyme to a high concentration of the correct substrate. So in this example, whether rubisco is inhibited by oxygen or not, we need to expose it to a high concentration of CO2. When you expose the enzyme to a high concentration of the substrate, it will actually cause the inhibitor molecule to unbind, and that's when it's a reversible inhibition, which is temporary. If nothing is changing nothing is causing the molecule to unbind to the enzyme, that is a permanent inhibition. This is when the bonds between the inhibitor molecule and the enzyme are so strong that nothing can break it. So that is considered a permanent inhibition that can't be reversed. So if you see a sudden change in the rate of enzymes, that looks like it's not coming back. It looks like it is going down in a very strong trend. It is more likely going to be a permanent inhibition, because with reversible inhibition, there might be a sudden recurve or a sudden, what's the word I'm looking for? Slanting of the reaction rate that could show that some of the enzymes are actually decreased. So essentially, yeah, let me just draw it out. So if you see suddenly reaction rate or and then reaction rate, and then imagine this is CO2 availability, right? So if you suddenly see reaction rate is dropping like this, you could, it's not really, this graph doesn't make sense because carbon dioxide, essentially if this started at 150 here, so on, right? If you suddenly see a very sharp drop like this, you could say that there are other factors playing, but another indication is that there's permanent inhibition occurring. That's why regardless of how much CO2 is present, there is no change in the reaction rate. Whereas if you see something that recurves like this, it could show that this is temporary inhibition, where when the CO2 concentration reaches a point, all the inhibitor molecules unbind to the enzyme. Chances are you won't see this graph. So I don't even know why I'm talking about this, but you need to be aware of reversible inhibition versus permanent inhibition. One will create a very obvious trend, and the other one will look kind of confusing. So you need to be able to differentiate that. So essentially, when we're thinking about enzyme inhibitors, many of the herbicide chemicals are actually inhibitors of enzymes involved in photosynthesis. For example, the first stage of photosynthesis, the light-dependent stage, can be inhibited by these kind of herbicides: uracils, triazymes, benzodiazepines. I'm not going to try. Okay. Any questions about this? Essentially, the presence of inhibitors will lower the rate of reaction, and as enzyme inhibitors can target enzymes within any part of both stages of the photosynthesis, all three types of plants are susceptible to the negative impacts of inhibitors. As you can see here. That's why my graph didn't make sense. Needs to go this way, not. So substrate concentration, you can see that what happens when there's competitive inhibitor versus non-competitive inhibitor. So non-competitive inhibitor is when it binds to any part of the enzyme known as the allosteric site and then changes the shape of the active site to become non-complementary. Whereas competitive inhibitor is when it binds to the active site and blocks the active site from allowing substrate to bind to it. But you can see here, this is the reaction rate if there is no inhibition at all. So you see that is showing a very sharp increase of reaction rate until reaching the saturation point, and then it plateaus. When you look at the competitive inhibitor, it is slower, but it eventually reaches that point as well. Who can explain why that is the case? Why is this non-competitive inhibition stuck at a very low reaction rate, whereas with competitive inhibitor, eventually it reaches the same reaction rate as as the uninhibited reaction rate? Who can give an explanation of why eventually competitive inhibitor reaction rate reaches the same point? It has to do with what I mentioned in regards to how can inhibition be reversed? If it is a reversible inhibition, what causes it to go back, reverse the inhibition? What is the factor that influences? I'll give you a hint, it's already in the graph. So remember, when it comes to inhibition, especially if it's temporary inhibition, the one thing that can cause the inhibitor molecule to unbind to the enzyme is substrate concentration. So if this is talking about photosynthesis, and we're talking about oxygen being the inhibitor molecule and CO2 being the substrate, when oxygen binds to the rubisco, it will slow down the reaction rate, or it will lower the reaction rate by promoting photorespiration. But as the substrate concentration increases continuously, more and more of those inhibited rubisco will unbind from oxygen and allow CO2 to bind to it instead. That's why the reaction rate continues to grow at a steady rate until it reaches the same plateau point where the concentration of CO2 is so high that no reaction could. All the rubisco is already binding to the CO2 instead of oxygen. Okay, so essentially what can reverse an inhibition is substrate concentration. The higher the substrate concentration, the lower the likelihood of the inhibitor molecule binding to the rubisco. Yeah. Moving on. Any questions about this? Anyone still confused about this graph and why it reached the final point? Anyone? Okay. Moving on. So this is a summary talking about each factor and how what happens if photosynthesis increases the factor, what happens to photosynthesis if you decrease the factor, and also how it can be different in C3, C4, and chem plants. So obviously, like if you increase it, it will increase the rate of reaction. But like any other factor, there is a sorry, not any other factor, but like a few other factors like carbon dioxide concentration. Yeah, these two factors have a saturation point. Okay, meaning a point where it's no longer gonna increase the reaction rate, it's gonna plateau. Right? Increases photosynthesis rate until a plateau is reached. You can see the same thing here for carbon dioxide concentration. When you decrease it, when you decrease light availability, obviously there will be a decrease in photosynthetic rate. Same thing with carbon dioxide concentration. Both of them will reduce in the photosynthetic rate. So when it comes to light, it's the same among all plants, meaning that all three plants, C3, C4, and CAM plants, are affected when there is no sun, there is light. So they're equally affected by light. So with temperature, increasing it to above the optimal level will cause the enzymes to denature. Decreasing it below its optimal level will cause the enzymes to freeze, essentially decreasing the rate due to fewer enzyme substrate collisions, meaning because the enzymes are starting to freeze or starting to vibrate lesser, they are more unlikely to actually bind to the substrate through enzyme substrate collisions. So when it comes to how temperature affects C3 and C4 and CAM plants, it's affected in a similar pattern, with C4 and CAM plants being better suited to hotter environments compared to C3 plants. That's part of their adaptations to survive in hot and dry environments. With pH, there is an optimal level. If it goes above the optimal level or if it goes below the optimal level, denaturation will start to happen, lowering the rate of reaction. And all three types of plants are equally affected by this. With water, essentially, if there's excess input of water, typically in excess in water, still as an input, we can say that increasing it will increase photosynthesis by avoiding closing of stomata. So the reason why water availability affects photosynthesis is directly related to whether it's closing or opening the stomata, and also as a reactant of photosynthesis. But you would mostly reference it as directly influencing whether it's opening or closing its stomata. If it has enough water, then it's going to keep the stomata open. Sorry, if it has excess water, it will keep the stomata open. If it has too much, if it's lost too much of its water, then it will close its stomata, and that will essentially trigger photorespiration at the end of the process. C4 and CAM plants are less impacted by changes compared to C3 because of their adaptations to consistently expose rubisco to CO2. So they are not really impacted a lot by water availability because they don't need to worry about whether the stomata is opening or closing. Essentially, they get to continue on with photosynthesis with their constant supply of CO2. And lastly, enzyme inhibition. So if there's too much inhibition happening, then the photosynthesis rate will decrease a lot. If there's fewer inhibitions, that essentially increases photosynthetic rate. And all three plants are equally affected by this.